Good morning. Welcome to day 2 of the San Antonio Breast Cancer Symposium. I have the honor of introducing our plenary speaker for today, Dr. Melissa Davis. Dr. Davis is the director of the Morehouse School of Medicine Institute of Translational Genomic Medicine and executive director of the Center for Genomic Diversity and professor of microbiology, biochemistry, and immunology at Morehouse in Atlanta. Dr. Davis completed her undergraduate work at a historically black college, Albany State University in Albany, Georgia, and obtained her PhD in molecular genetics at the University of Georgia in Athens. Her postdoctoral training was a functional genomics and systems biology, which led to her focus in racial disparities of breast cancer mortality and using genetic ancestry tools to identify biological determinants of these disparities. Her pioneering work has uncovered groundbreaking discoveries in genetic networks related to immunological and tumor phenotype distinctions across patient groups from African ancestry. Earlier this year, a global team led by Dr. Davis was awarded a $25 million grant funded by Cancer Research UK and the National Cancer Institute through Cancer Grant Challenges to address cancer disparities in populations of African ancestry, becoming the first Cancer Grant Challenge awardee to focus on cancer inequities. And this grant is the first one that is awarded to an investigator at a historical black medical school. Please join me in welcoming Dr. Davis to the podium for her lecture titled, The Grand Challenge of Unraveling Social versus Biological Drivers of Racial Disparities in Cancer Outcomes. Dr. Davis?
Thank you. All right, good morning. It's early. First of all, thank you to the organizers for inviting me to talk to you about our work. My quick disclosures. All right, here's our outline for the discussion today. We're going to start with a brief background of the emergence and persistence of disparities. So, how were disparities originally defined and observed, and what factors led to these disparate outcomes to begin with? And more importantly, why have they persisted despite decades of effort? What we already know is that racial segregation in the United States leads to a segregation of socioeconomic status. Shown here is a map of the United States and county by county, the percentage of poverty in the United States. Now, as I overlay where African Americans live, you can see that there's a clear correlation of this minoritized population with socioeconomic status. Landmark papers, starting in the early 80s, the NCI initiated studies to uncover what they called explanatory factors of the black-white disparities in cancer survival. What we see here is a series of hypotheses that centered on socio-behavioral indicators, which led to a series of landmark papers that basically reinforced that socio-behavioral factors were the key driving force of the disparities that we saw in mortality. But is it only SES? Is it only social determinants? So let's revisit the map. African Americans in the United States are not a monolith. We've actually overcome some of these SES barriers. And now if we can re-stratify based on not only race but socioeconomic status, what becomes very obvious is that as African Americans move into affluence, this does not improve their outcomes. In fact, as we look at breast cancer outcomes, which emerged only in the early 90s, black patients who live in affluence still have a higher mortality than white patients who live in poverty. So this tells us that SES status isn't the only factor that's driving disparities. In fact, at the emergence of the disparity, we know that certain advents such as endocrine-targeted therapies were what created this stark decline in the white population that didn't replicate in the black population till nearly a decade later. So we know that factors that cause disparities are multifaceted and ever evolving with an evolving societal landscape. The key to understanding how we might overcome disparities is through systematic deconstruction of these interwoven factors and actually measuring how much of an influence each one of them may have across the entire cancer care continuum. In a study led by Erica Warner at Harvard, what she helped us understand is that tumor characteristics actually plays a very significant role in racial disparities in breast cancer. However, interwoven into this tumor characteristic is, of course, the fact that Black patients still have negative experiences throughout their cancer care. But the tumor characteristics that are most obvious start with hormone receptor status. In the United States, we know that triple-negative breast cancer is more than twice as likely to be diagnosed in Black patients. But we also know that some of the disparities that we see, my initial studies in disparities with Fumio Lapade at University of Chicago, showed that the replication of these race group disparities was actually driven by ER-negative status in this population. But this was also our first insight into biological factors, where a cofactor for estrogen receptor was differentially distributed within the cellular structure of African American and West African women. In a more recent study with Dr. Lisa Newman, where we took about 20 years worth of data representing Detroit and the New York City area, what we were not expecting to find was this wide gap in disparate outcome between screen and non-screen detected triple-negative breast cancer patients in African Americans, where the expected difference in survival more than doubled in this particular population. And this is despite the fact that white Americans in this population had twice the number of non-screen detected cancers. And across the top, what you can see in the pie charts is that there wasn't a significant difference in the stage distribution. And so while these patients were being treated by the same doctors in the same healthcare system, what this tells us is that the treatment doesn't work the same. And why would that be? Is there a genetic risk here that we haven't really considered? Is there an incompatibility, for instance, to the standard of care, which at the time was systemic chemotherapy? What I'm showing here is known pharmacogenetic alleles that are related to cardiotoxicity to anthracycline therapies. And we have emerging findings coming from young investigators such as Arnetha Sutton, which are helping us to understand how these racial disparities in treatment-related cardiovascular toxicities may also be driving some of the disparities that we see in outcomes. But what's clear here is that there is a bias in the distribution of this allele, where nearly a third of the African and African diaspora have this particular predisposition for cardiotoxicity, but it currently is not a consideration in clinical care. Yet the controversy of ancestry versus race persists. So this is a small drama that started to emerge in the literature. I won't take a lot of time here, but I will say that there is still a bit of tension about utilizing genetic ancestry as opposed to the very strong notion that socioeconomic status and racism are the only factors that need to be studied. But the reality is we don't have enough data about genetic ancestry to make that assumption. If we look at the knowledge gaps that we've created by sampling bias in GWAS studies, for instance, we find that, again, the majority of the data that we have for GWAS studies was originated from people of European ancestry, which led to inaccurate clinical tools, where here you also see that if we study the genes, the cancer genes of any other ancestry group than European, there's a higher likelihood of finding a variant that we've never seen before. So this higher rate of variance of unknown significance is a clear indicator of the inaccuracy of clinical tools. So how do we overcome these limitations? By including diversity in not only our cohorts, but our investigators. In a theory that was coined by Dr. Lisa Newman, oncologic anthropology, we use this global oncologic approach to really think about triple-negative breast cancer across the world. What we found was that the highest rate of triple-negative breast cancer is in sub-Saharan Africa, and then in every nation where we can separate the population by self-reported race or ancestry, it is the Black component of the population that has the highest rate of triple-negative breast cancer. We even measured West African ancestry in our multi-ethnic population of breast cancer patients and found that West African ancestry was associated with a higher likelihood of having triple-negative breast cancer. And this was the first time that we were able to show that ancestry had a significant influence on the subtype. But what does that even mean? If I say that ancestry in an admixed population is associated with a certain phenotype, how does one interpret that? We know that admixture, where you're from, is stochastically painted across your entire genome. Depicted here are several chromosomes that represent our African diaspora, and it can be a range of West African ancestry from as low as 10 to as much as 90%. But what we're looking for is a region of the genome that is shared with West African ancestry, and it looks a bit like this. So as we scan West African ancestry and local ancestry studies, what we should find in triple-negative patients is a spike that represents this shared ancestry background, which then allows us to hone into what is yet still an unknown allele that provides this predisposition. And let's think about the African diaspora. So population differences and gene mutations is well known. We know it through a founder mutation, and it's actually how we define our different human populations. And within this geographic distribution, there are certainly alleles that have clinical relevance, shown here, for instance, the geographic distribution of the blood group alleles for A and B blood types. But these were the result of founder events. How much more will the influence of West African ancestry across the transatlantic slave trade then also carry with it the predisposition of these African genomes, where millions, 13 million Africans were displaced over centuries? And so what we're seeing here is a population level event where we can actually see the influence of genetic background by way of lineage, which is West African ancestry. And so here's the grand challenge: to incorporate all of this prior knowledge into a study that will allow us to unravel all of these different factors. So meet my team, SAMBI. SAMBI is, as was in the introduction alluded to, the first grand challenge team that was funded for cancer inequities. Our team is made up of 5 different work packages, which I'll introduce to you here. Our logo is a lens, and the aperture of this lens incorporates all of our different disciplines, and it's focused on the African diaspora. In our study design, our target population is 40,000 enrollees, including cases and controls, which incorporate oversampling of African ancestry, and we're focused on 3 different cancer types, including breast, prostate, and pancreatic cancer. As we think about how we wanted to tier each one of the work packages, we put social determinants of health right at the foundation. So as we move up in tiers through exposomics, genomics, and immune response, this will be a historic collection of comprehensive data across the same set of patients, because really that's the limitation and the frustration that we're trying to overcome, where each one of these disciplines has done marvelous work, but only in silos. Social Determinants of Health work package is led by Lauren McCullough and Tiffany Carson, and their goal is to reframe the tools and measurements of social, societal, and political determinants of health. What we understand is that African Americans and the Black population in general across the globe is not a monolith. And so measures that are currently the standard, such as neighborhood deprivation indices, don't actually capture the full range of what is impacting the health outcomes of this population. In fact, Dr. McCullough found that neighborhood deprivation indices don't reflect the outcome, especially in the highest SES strata, where in Atlanta, the top 1% and 2% earners of our African American population are 126% more likely to die of breast cancer. So as we consider societal impacts, including trauma from, for instance, across our African nations, civil wars, natural disasters that displace people and have entire villages living as refugees, we will have a multi-layered approach of not only incorporating the area-level social determinants of health, but also the socio-cultural and individual lived experiences. What we know very much so in the United States is how redlining, which is a systemic racism effect of government-sanctioned city planning and disallowing certain ethnic groups to live in certain regions of cities, is also overlaid with, for instance, the overexposures of hazardous waste, which correspond to cancer disparities. So what I'm showing here are the oversaturation of health hazard sites within the boroughs that are imprinted across these historic redlining events. And so it's important to really fully measure and understand these exposures, which brings us to the second work package of exposomics, led by Dr. Gary Miller and Dr. Zola Dlamini. Now, we have some known factors through our epidemiological studies where we know that the exposures of certain mutagens in our environment are causing, for instance, the etiology of specific tumors. But what we haven't been able to fully uncover are the diversity of unknown factors. Unknown factors that, although the source may be different across the diaspora, the particulates may actually be the same. So in this work package, we will proceed with a series of mass spectrometry readings on plasma, which will give us over 200,000 data points, which I'm probably underestimating. And Dr. Miller has already successfully been able to identify particulates that were unique to ethnic groups in an Alzheimer's study, where he showed that, for instance, Hispanic, Caucasian, and African Americans living in New York City had unique particulate patterns or signatures that were related to network pathway analyses. He also showed that these particulates were unique to the boroughs that they lived in and was associated with a more severe form of Alzheimer's disease. He was able to even link it to where they lived being in proximity to the MTA bus depots, which was why they were being overexposed by some of these particulates. You may have heard of one of them, PM 2.5. And how much more impactful will this type of study be then in countries in Africa where, for instance, air quality isn't even measured at the rate that it is here in the United States? Which leads me to our genomics work package, where we hope to have over 40,000 germline whole genome sequences and 4,000 long-read and transcriptomics on tumor and normal samples across our population. Our team leads are displayed here. Now, we're standing on the fact that we've been looking at genomic signatures for quite some time. So in reanalyzing, for instance, the Cancer Genome Atlas data about 10 years ago now, we were able to show that in triple-negative breast cancer cases, African Americans were more likely to have a quadruple-negative cancer, where androgen receptor negativity was also present in over 80% of African Americans. It was an even earlier onset than triple-negative breast cancer. We also looked at, for instance, omics using microRNAs and found a unique microRNA signature that was specific to African Americans with quadruple-negative breast cancer, one of which is a regulator of the MYC pathway, but it was only relevant, clinically relevant in African Americans. Moving on to studying cohorts that we built ourselves in collaboration with Dr. Clayton Yates, we at the time were only able to use self-identified race. But as our methods became more sophisticated and we started to utilize ancestry, the dichotomy of gene expression became much clearer. In fact, we were able to show that even in an admixed population of European Americans, there's a gene signature that is reflected differently because of their genetic admixture. Now, this work was amplified even more at the New York Genome Center at the onset of the Polyethnic 1000 project, which is the vision of Dr. Harold Varmus and others there. It was a collaboration across the cancer centers across the New York City area, trying to harness and leverage the diversity of what we used to call the melting pot and we now call the tossed salad of New York. We were able to see and publish a landmark paper in cancer discovery by the optimized pipelines from the P1000, where we utilized genetic ancestry in a different way as a continuous variable. And this allowed us to stratify our population and identify even a subset of genes that reflected a true dichotomy of expression depending on the genetic background of the gene. This tells us that there are ancestral informative alleles that are driving the expression of networks of genes. And so based on that set of findings, we'll start to expand our work in Work Package 3 and hope to find even more ancestry specific, but maybe even other types of factors from, for instance, Work Package 2 and Work Package 1 that help us understand how differentially expressed genes in tumor biology reflects this unique outcome. But one of my favorite deliverables from this work package is a set of human genome references from the Pan-Genome Consortium that will be reflective of this unique diversity of the African diaspora. Some hints to the influence of African ancestry are in mutational signatures that are associated with ancestry, as well as mutational signatures that are emerging to be associated with the African version of certain DNA repair genes. We're even able to display that the utility of using a pangenome reference also allows us to identify novel regions of the genome where clearly there are yet to be discovered factors in our gene expression signatures that we've never interrogated before. We have regions of epigenetic regulation where we've identified over 6,000 contigs with novel CPG islands that have never been interrogated before in the dark genome, if you will, as well as thousands of what was previously unmappable gene expression reads. But one of the things that also rings true is that there's still a subset of genes that are strongly associated with race that are not associated with ancestry. And when we look at what these genes actually reflect, they are the comorbidities in this population. And how then will this impact tumor biology? Which brings me to our fourth work package. Standing again on the work that we had already published showing using deconvolution that ancestry was associated with a higher infiltration of immune cells, which we showed using RNA-seq as well as IHC, we were able to now start to think more clearly about the status of these immune cells because it was counterintuitive. How would ancestry give us a stronger immune response, yet in the context of having a lower survival? So what we see here is that African Americans completely lose the survival advantage of having a higher infiltrate of immune cells. We believe that one of the reasons is because of another blood group called Duffy null. This mutation was fixed in sub-Saharan Africa because it confers malarial resistance, but we also know that the expression of the gene in the context of tumor biology leads to the infiltration of immune cells by way of its activity of regulating cytokines. We also showed that the gene expression in tumors was associated with better overall outcomes in breast cancer. And so now in over 1,000 patients, we've been able to study the allele itself and have identified a signature that is African American specific that correlates with a unique spatial tumor immune profile that we believe is going to be even more amplified as we expand our cohort to a global population. And stay tuned for the dark side of breast cancer. And so as we start to integrate the data from the work packages before work package 4, we call this population our platinum cohort because it will be the first time we'll have comprehensive data from social determinants, environmental exposures, whole genome and transcriptomics, as well as single cell spatial information and circulating inflammatory signatures and immune cell profiles to help us untangle what are the more influential factors leading to this tumor biology. But the translation of this work into clinical practice is crucial. If we think about certain trials where they did at least try to track genetic ancestry by way of self-reported race, in the Impassion 130 trial, for instance, where they showed PD-L1 was an important factor and their immunotherapy drugs showed better overall outcome in basal-like immune activated cells, tumors, we see that African Americans actually had the strongest response, but the number of cases were so low that we weren't able to make any clear delineation. But this is where our patient involvement work package comes in. It will be the patient voice that will help us to translate this, and led by Ricky Fairley, who's been able to enroll over 20,000 African American women in clinical trial registries over the last 2 years alone. We know that with her vision, we'll be able to not only disseminate the work that we'll be doing here, but create policies and advocacy for equity in cancer care. One of our deliverables is to create a standby library. For every scientific paper, we will publish a plain language version for the community. And our vision, helped here with a bit of AI, is a community library for bidirectional conversation. I want to leave you with the promise of integrating these world-class methodologies. As an example from our BCRF study, where we are not only looking at spatial differences in tumor, but geospatial differences in location. As we look at the underlying redlining and where each one of our patients live in the Detroit and New York City area, as we combine whole genome sequencing markers for instance, in this case, I'm going to show you the mutational signatures, and we also start to incorporate circulating biomarkers from exposomics. As we incorporate information about the environment, we hope to start to identify how where people live is a reflection of mutational signatures in their tumor. Here, showing, for instance, in the boroughs of New York, the signatures incorporated with not only the mutational signature, but area level deprivation, and even showing sort of an inflammation, if you will, of certain signatures in regions of Detroit. We hope that this will also allow us to fully appreciate how, in the circle of precision medicine, geography, ancestry, the lived experience, not just socioeconomic status, will be a reflection of the diversity that's necessary to truly create the precision medicine that's necessary to overcome inequities. And with that, I'd like to thank my teams and our funders. And I'll leave you with a quote from Dr. Harold Freeman in his commentary on the meaning of race and science and society, all the way back in 2003. What he says is, we seek scientific truth, but that drive must always be wedded to concern for social justice. Even in the lab, it's not enough to simply find an answer and go on to the next question without considering how our discoveries can affect real people in the neighborhoods throughout this country and the world community. Thank you so much for your attention.
Thank you. Thank you, Dr. Davis, for that most informative presentation.